The next two speakers are Dr. Emmanuel Cummings and Dr. Joseph Lee. Dr. Emmanuel Cummings is Deputy Vice Chancellor, Academic Engagement, University of Guyana. He has extensive experience in university administration and pioneered the transformation of the College of Medical Sciences, UG, while serving as the Dean. He is a Fulbright Scholar, full member of the British Physiological Society, and the Health Systems Global, an eminent medical researcher. His research focuses on nutritional management of diabetes, cancer, and eye care. Dr. Joseph Lee is a genetic epidemiologist with extensive research experience in conducting integrative genomic studies of aging, Alzheimer's disease, AD, and AD-related traits, and several unique high-risk cohorts in different parts of the world. The focus of his research is to map genes that contribute to Alzheimer's disease, AD, and related traits to better understand the relations among genetic factors in AD, as well as other AD-related phenotypes, but applying statistical genetics methods. Dr. Lee is also a co-investigator on the Guyana Well-Being Study examining sociocultural and genetic factors underlying suicide behaviors in Guyana.
Good morning. Thanks for the invitation, and I'm happy to be here. So today I'm going to talk about genetics of aging and age-related diseases, and mainly it will be Alzheimer's disease as an example of studies that I conduct. So I'm Joe Lee. I'm at Columbia University, and you can see the rest. So before I forget, I just want to acknowledge some of my collaborators. These are my team members. Dr. Schiff is my Down syndrome collaborator, and Dr. Meyer is my Alzheimer's collaborators. And Christian Hogan is a collaborator on Guyana project. Andrew Sproul and Catherine Marquer are cell biologists that, who I collaborate with, and these are my Down syndrome collaborators, and these are collaborators at University of Puerto Rico and University of Zulia in Venezuela, and Dr. Maestri and Dr. Medrano in Dominican Republic. So I have various projects across North America and Latin America, and some projects in Kazakhstan and Finland and so forth. And these are the funding sources that made this possible. And plug for a web-based course that my colleague and I conduct through Columbia Global Center. So this is a web-based course on human genetics, and it's for young scientists who wants to learn something about human genetics. We don't talk about technical details, but rather we talk about how and why we should study human genetics in various diseases. And it's this is the website at the bottom, and it happens once a month. So if you choose to, if you have nothing better to do, and you can attend this course, web-based course, and it's free. So that should be attractive. So let me start with aging the basics. So it's been shown that over time, the human life expectancy has increased, and in the Stone Ages, the average 50% of the people lived to age 10 here, and then when it became 1840, that increased to near 50 years of age, and then by 1940, that increased by another 15 years or so, and by the time in current days, the life expectancy across the Western country or globally is around 80 years of age right now. But there are a lot of variation across different countries, as you can see in this global map, that where you see green, and these are the places, countries that have a longer life expectancy, and then you can see that it's North America, and then the European countries and Australia, and South America is doing reasonably well, and Africa has some catching up to do. So this is the life expectancy in Guyana. According to the statistic that I got, at the moment, the life expectancy of a Guyanan population is about 70, and compared to other countries, it's got some catching up to do. But as you can see, that there is a slight trends for increase every year, and the reason for slight increase is because that annual changes is about 10%, a little higher than 10%. That's why you see only a small increase over time. So is that a good thing? Because people are living a longer period of time. As you can see that as people's life expectancy increases, then what's going to go up is the prevalence of age-related diseases. And as you can see that as you get older, things like arthritis and other things, other diseases, age-related disease will go up. And coronary heart disease, of course, goes up as you get older. But you notice that diabetes and cancer, the disease prevalence actually goes down because people die. If you have these two diseases, you're going to die before you reach age 8 years old. So that's a different way of looking at it, to see that some things will go down as you get older. The chances are going to get decreased. So if you were to eliminate all cancers from the human medical term, you will see on average increase in 2 years in life expectancy. And obviously heart disease is going to have slightly more. And this is a calculation based on Sergievsky in 2001. So since the title, as the title says, I said genetics of aging and age-related disease, I'm going to talk a little bit about why study genomics. Well, the reason to study genomics in understanding longevity or aging is the following. Environmental risk and lifestyle factors are likely to be the main factor. But if you look at the heritability of longevity, it is estimated to be about 20% according to a twin study. But when you look across different population, the increase in life expectancy, you see it all over many countries, and you see the global change in the trend. So if you see such a wide range of increase, that is highly unlikely to be due to genetics. It's more likely to be environmental change and changes in medical care. That's why you see an increase all over the place. But that doesn't mean that genetic factors are not important. What's going to happen is that there are many genetic factors that contribute little to each aspects of longevity. And in the following, I'm going to show you a simple model and then to more and more complex models. So in the modern day medicine, this is what you see when you turn on the TV or open a newspaper. You're going to see that you see the things that says, oh, scientists just doubled the life expectancy of fruit flies. And then you will say, oh, who cares? It's just fruit flies. But it gives you some idea that one can manipulate a genome to change the way your body is behaving. So to that end, England has started in 2007 what's called UK Biobank, where they start collecting biosamples from about 500,000 Britons, citizens. And then because they have national health, they were able to connect the two biomarker data with their medical records, and they are able to generate incredible amount of medical information. So that right now, if you are a qualified scientist, you can actually use their data to understand how things are related between gene and the Alzheimer's disease or heart disease, what have you. And not to be outdone, United States, they have a program called All of Us, where the goal is to sequence about 1 million people and then link their medical record in the hospital to understand how genotype and phenotype relationship it is in American population. And Columbia University is one of the sites in this multi-center effort. And then as here, Francis Collins, the head of the National Institutes of Health, has projected that in the future, when you take the drop of blood from your patient, you may be able to see and predict who's going to have what disease and when, and which I believe is far too optimistic in projecting such power with a modern medicine based on what we know and how complicated the diseases are. So once again, why study genetics? Well, I study genetics because that's what I'm trying to do. You know, when you're a hammer, when you see a nail, what you do is you want to nail it down, and that's what geneticists do. When you see any diseases, you want to find out how genes affect the outcome of the disease. And as you can see in this picture, on the left of that hammer picture is the tall man is Will Chamberlain, who's one of the best basketball player in history. And the other one is Schumacher, is one of the best horse race jockey in history. So as you can see, these people are very different, but they are very genetically different. And then they display a wide range of height difference in humans. And then, so if you were to look at longevity as a trait that you want to study how genes influence their outcome of longevity, it has been studied based on twin study that about 20% of the variation in longevity is due to genes. And when you look at the age-related disease like Alzheimer's disease, approximately 50% of genetic variation is due to Alzheimer's is due to genes. And in most common diseases, there are many genes involved, and each gene is likely to contribute a small effect. And then one gene interacts with the other genes. So those are modifier genes. So the process is quite complex. And as I said before, the predictability of single gene is going to be weak to modest, unless you happen to have very rare Mendelian disorder, where if it's inherited in autosomal dominant recessive pattern, you are likely to have Alzheimer's disease or whatever disease you have. But I'll tell you even complications in those studies further. So because there is only small effect of genes on the outcome, what we want to study is you want to study high-risk people, because this is where it's easy to observe what is a genotypic impact on the phenotype is. And so one of the studies that I do is looking at Down syndrome individuals because they have 3 chromosomes on chromosome 21 that causes Alzheimer's disease. And when you look at those people, and then you see that most people start showing neuropathology at age 40, but if you look at them long enough, and then you realize that there are people who are completely resistant to Alzheimer's disease, and that the question maybe you may ask is that what kind of things that prevents them from developing Alzheimer's disease? So there may be a genetic protective factors these people may carry. So studying high-risk population is an interesting way to flip the coin to ask the question from, yes, you have high risk, how did you manage to escape the disease? And that's another research question that we can look at. So let me walk you through some of the lower animal models that has been done. In late 90s, the professor at UCSF Medical School took the C. elegans worm and then tweaked the gene called DAF2, and then by knocking in certain mutations into that C. elegans, she was able to increase the life expectancy of that worm by 2-fold. So, and then when they sequenced this gene, it turns out that the gene sequence was very comparable to human insulin receptor genes. So then they studied further, and then it was shown that it is associated with stress resistance, metabolism, and the developmental stages. And as you can see, it's related to various antioxidant processes as well as immune responses, and that may lead to, in some ways, longevity. And these signaling pathways that they were able to observe in C. elegans was comparable to mammalian insulin pathways. So another example is single autosomal recessive disease called Werner syndrome. And this is adult progeria, where the incidence of this disease is about 1 in 100,000, and it happens 1 in 40,000 in Japan, and then 1 in 50,000 in Sardinia. And the reason it's twice as high in Japan and Sardinia is because there is a marriage among family members that allow the frequency of those mutations to be more common in these subset of population. It's a certain part of Japan, not Japan as a whole, that has elevated frequency of these genes. And then this gene has been identified, and it's in the short arm of chromosome 8, and it follows autosomal recessive pattern. And this person was, when she was a teenager, this is how she looked on the left. And by the time she reached age 48, she's rapidly aging such that she looked like somebody in her 70s and 80s. So this shows incredible rate of accelerated aging in terms of every aspects of that person's physiology. And so that I just showed you 2 genes that appear to be working against aging. And then subsequent studies have shown that there are genes that seems to be promoting healthy aging, so that when the group of scientists examined the people who lived to 100 years old, they found that the frequency of FOXO3A variant was about 40–50% in these people, especially among the people who survived to really old age. And then you can see in the picture on the lower right that they have highly expressed FOXO3A protein in these individuals who are survivors to old age. So this table summarizes some of the how genetic certain traits are. So as you can see, type 1 diabetes is highly genetic, so that the heritability estimate represents how genes contribute to the trait. And the maximum number is 1.0, so that 9.9 means 90%. And then if you look at bipolar affective disorder, that's about 60–70% have some genetic contributions. And whereas 80% of the contribution may be height in their determination of how tall you're gonna be. So does this mean the genes dominate what you're gonna be likely to have these disorders? No. I'll show you in the following slides that this is not, you need to take it with a grain of salt. So, and then let's think about, if you are gonna think about how genes may contribute to aging process, Then there are some of the scientific questions that you can ask is, why do we age, and how do we age? And then how do we measure aging? And then what is normal aging? And then when we age, why do we have an increased risk of certain diseases like heart disease and cancer and Alzheimer's disease and so forth? So if aging is just breaking down of your body parts, why does that make you more susceptible to having these chronic diseases? So these are some of the questions that we need to address. It is most likely to be, it's the nature-nurture thing. Some of it is going to be due to genes, and some of it is going to be due to environment. And each population that you're going to study will behave differently. And there's no one fixed rule that something's going to happen one way, but not the other. So I'm going to introduce you to complexity in aging and age-related diseases. So genetic variants alone do not predict the disease risk. And of course, environmental factors do not necessarily predict disease risk either, because if you've read enough papers that says salt increases hypertension and so forth, but you look around, then there are people who eat very salty food and that are perfectly fine and then normotensive and so forth. So there are lots of variation across people. And then, so most likely it's going to be interaction between gene and environment. And then if there is a gene environment interaction, you have to figure out, well, which gene is it that is interacting with which environment to understand this disease processes. And there are over 25,000 genes to deal with. And then there are 3 billion nucleotides in the genome that can potentially contribute to genetic variation. And then, as you know, environmental factors can be billions, and you have absolutely no idea which environmental factor and what stage in life environmental exposure may have the biggest impact on your disease outcome. So let me take a simple example of Alzheimer's disease. It is well known that if you carry ApoE4 allele, then you have increased risk of Alzheimer's disease. So studies in Washington Heights at Columbia, we did this study. I wasn't involved in this study, but it was determined that if you're a white population, if you belong to white population, and then if you carry ApoE4 allele, then your risk of Alzheimer's disease goes up by about 3-fold. However, if you're African American, there is essentially very little increased risk in Alzheimer's disease if you're carrying ApoE4 compared to those people who do not carry ApoE4. So, and then Hispanics, there is a slight increase in the people who carry ApoE4 compared to those who do not. So the risk associated with ApoE4 varies highly by what genetic background you belong to. But in the same paper, they calculated absolute risk of Alzheimer's disease across age. As you can see, that this is the risk of ApoE4 carrier among whites. And then this is that for those who do not carry. As you can see, there is an increase, and these are the white folks who has increased risk of 2.5-fold if you carry ApoE4. However, if you compare those risks to African American who carry ApoE4, you can see that they, even though African American, their relative risk in E4 carriers compared to non-carriers is trivial, you can see that their absolute risk of developing Alzheimer's disease is very high. So when you read these genetic literature, you have to be very aware of, is it absolute risk they're talking about, or is it the relative risk they're talking about? Because if they're relative risk they're talking about, you have to always look at the reference group that they're comparing against. Now, this is an example of genotype-cohort relationship. And then it is well known that gene called FTO predicts who's going to be obese over time. So this is age across time, and then this is BMI. So if you look at the red line, red solid line represents people who carry a genotype of this FTO gene, and then the people who are in the dotted line are the same people who carry a genotype. However, this is after World War II, and this is before World War II. So depending on after World War II, people's diet changed so that they consume a lot of fatty food and more calorie than pre-World War II, so that you can see that even if you have a same genotype, depending on what you eat and how you behave and how your lifestyle factors affect you, you are going to see even the people with the same genotype have a strong impact on their outcome of obesity phenotype. And this graph just illustrates how your risk of various disease changes over time. Some diseases, it goes up, and for some diseases, it goes down, and this all depends on who survives to a certain age and beyond. So if you look at this cartoon, what this tells you is that if you want to study whatever Alzheimer's disease, depression, heart disease, whatever you are thinking of studying, there is a major gene that you want to be focusing on. However, there are lots of other major genes that you need to take into account. However, and then familial level of genetics also contribute to this whole genomic milieu that this person is experiencing. And then there is a population level genetics. So that when we are looking at people in Washington Heights, in Manhattan, you'll see that there are people from 190 different countries all living in the same place. So that when we say genetics, we have to be careful as to what genetic genes, genetic factors we're talking about. Is it the gene itself, or is it whether the family came from certain part of Italy, like Sardinia or something, or whether your ethnic background is very different, as I showed you 2 slides ago about the difference between the white, black, and Hispanic. So there's a population genetic, familial genetic, and then the individual genetic factors. But at the same time, the same person is exposed to different environment. Maybe that person smokes cigarettes, and then the family environment, and that person is exposed to familial environment, and then their cultural background, because if they're Jamaican, and then they may be eating Jamaican food as opposed to Dominican food, and then they are living in Manhattan, so they're going to be exposed to Manhattan environment. So all these factors play a role, contribute to this whole array of phenotype, and this is really quite messy, but this is not enough. Because when you look at the disease, you can see that there are a number of subtraits that happen, and you see the direction of the arrows, and then having blood pressure is going to lead to some other trait, and that other trait is going to lead to some other trait. So the disease is going to be a culmination of number of different endophenotypes that happens in that person, and then to each of these steps, there's environmental and the genetic factor that contribute to it. So to understand these complicated, complex biological phenomenon, you need multidisciplinary efforts. So you have to study demographic, and then you have to study anthropology so that you know how culturally they're different or similar between different groups of ethnic background within the same population. And then you obviously have to see epidemiology and statistics to see what kind of risk factors each of these groups have. And obviously you need to have a good medical system that tells, oh, this person is having hypertension and that's going to be leading to eventually some vascular dementia or what have you. And then once you have all these things, then you want to see how genes affect their risk. And then once you have those genes and all these risk factors, then you want to look into systems biology to see how these multiple risk factors interact with one another. And then once you have a good idea of which factors are important, then you want to figure out cellular biology to see what at molecular level, how are these things happening? As you can see, to understand any one disease, it's a massive effort of multidisciplinary effort are needed to get this understood properly. Just to give you an example of genomics being a great thing, which I do agree that genomics is very important. However, this is a study published in Nature Genetics that says they examined 250,000 people, and then they discover 14 new genes that affect the obesity level, and then they had 18 new additional loci, genetic loci that affects BMI. But altogether, these 32 variants, genetic variants, explain less than 20% of the phenotypic variance. So just like that cohort effect that I showed you, you can have these 32 genetic variants, but depending on what your lifestyle is, what your dietary intake is, the likelihood of you being extremely obese or modestly obese is going to be highly variable. And to understand all these stuff, you need something called multi-omic approach. So in the olden days, when I was a graduate student, we used to study gene and the outcome, whether it be healthy aging and Alzheimer's disease. And then now all the technology has advanced to a degree that you can see every level of molecular changes, and then you can also see which direction they're going. So the degree to which we can understand molecular biology has advanced tremendously. However, we still have a lot to understand. So I'm going to give you one example of the study that I'm doing in Puerto Rico. So these people carry a gene that causes Alzheimer's disease in PS1, and I'm going to skip that. So what we discovered this mutation in 2001, and then this mutation caused high level of amyloid beta. As you can see, it's halfway between APP mutation and the wild type. And when we examine these families, we see that within a family, some families have early on, some people have family late onset, but within a family, there's a large variation in age at onset. We want to see what causes these variations. And we learned that environmental risk factors such as stroke and head injury and smoking did not explain why there is a variability. However, when we looked at physical activity, the people who are actively engaged in physical activity were 6 years more likely to delay their age at onset by 6 years or so. And the complication is that when you look at Puerto Ricans, some people have Caucasian background, some people have African background, some people have American Indian background. So the genetic background is very complicated. So we study the entire population, entire family to see how these things work. And we discover several genes right now. We are looking to see how they affect. So when we study Alzheimer's disease, we look at these high-risk people, and we see that when you have a SINJ1 mutation, it changes age at onset. And then we take their brain, and then in Down syndrome to see how SINJ1 behaves in their protein level. And then we have a knock-in mouse to see how that mutation affects their outcome. And these just illustrate some of the results. from our analysis, so that if you have this mutation in CINCH1, you're gonna be, onset is delayed by about 15 years. And then this shows from the brain that, and when you have these mutations, then your protein in your brain is going to be severely affected. And then that has been also confirmed in a mouse model. So this shows that when you are dealing with a high-risk population with a mutation, we ask, you should be at high risk of developing Alzheimer's disease. But some are not, then we ask, what is the factor that allow them to escape having Alzheimer's disease? So, but in order to do that, you need to understand genetic architecture to accurately assess their genetic risk. And then once we do that, we have to do a functional genomics work. And then this is my last example, where we are looking at people who lived 100 years of age. And if you have ApoE4, I know, as I told you, they're most likely to develop Alzheimer's disease. And then we are looking at all the intermediate level phenotype by examining their metabolomics. And then say here, if you have ApoE4, you should be, you're at high risk of developing AD compared to ApoE3. And then, is there any difference in molecular structure of these people in ApoE4, so that some develop AD and some do not? And then to do this, we examine families who lived 100 years old. And then we looked at about over 300 metabolites as how they're metabolically processing their biological system. And this shows that we came up with a 2 cluster of proteins that specifies who's going to develop Alzheimer's disease versus who's not going to develop Alzheimer's disease. And then once we discover which proteins are going to be associated with developing Alzheimer's disease, we look to see which genes are associated with their elevated levels of those proteins. So this is, well, these are the genes that we, potential genes that may explain some of the relationships. So these are candidate genes. So using these genes, now I ask my cell biologist colleagues to do some functional work that may affect the disease genotype-phenotype relationship. And I borrowed this from Mark Mapstone at UC Irvine, and this shows that when we talk about these genes and affecting Alzheimer's disease and otherwise, you see the number of genes involved. This is the biological pathway that can potentially explain how these relationships may happen. So to summarize, human variation is widely observed and genetics is highly informative. Common diseases are very complex, involving environmental and genetic factors, and they interact with each other. So if you have a different genetic background, if you have a different environmental factor, the results are going to be different. And in order to understand this genotype-phenotype relationship, you have to study your own population. For you, you have to study your Guyanan population. And as I understand, Guyana has several different ethnic background of Africans and Indians and various other ethnic groups. You have to understand how differently these populations have their genetic and disease risk. And to understand this, the best answer for Guyanan population, you have to do your own research. And I think that's why you guys are doing this symposium too. And then if you have a time, lastly, attend the web-based course. This will talk about something about how to do human genetic studies in a more conceptual level rather than a technical detail of, it's not a recipe course, it's a conceptual course. Okay. So that's all there is for me. And I spent 45 minutes, so hopefully Dr. Cummings may have some wisdom to share, and I'm done here. Hello?
Hello, Dr. Cummings, do you have, okay. And there's one question, someone wanted to know if you can explain a little bit more about the relative and absolute risk.
Oh yeah, so the difference between relative risk and absolute risk is this, you know, when you have a high risk of Alzheimer's disease in one population, you're compared to the people who are, you know, one genotype. That genotype person can have a relative risk of 10% of developing Alzheimer's disease. But if you have the other allele, and then if you have 30% risk of developing Alzheimer's disease, your relative risk is 0.3 divided by 0.1, so it's 3 times higher. So that's 3 times higher risk of developing if you have certain genotype in one comparison. However, if you look at ignoring all the genotype, if you start following people up and say, oh yeah, this person has, and that person may have a 50% or 60%, but because they belong to a different ethnic group, you may have a different reference comparison population, so that it all depends on what the reference population you're comparing against. So that's why if you're going to be doing genetic counseling, you have to be very clear about the risk you have to tell is a relative risk or absolute risk, because you know a genetic counselor who's working in Manhattan has to be very aware that yeah, you only have twice as high a risk of developing this when you don't have this mutation. However, that person may have 50% chance of developing certain disease by age 70. So you have to, from that person's point of view, excuse me, the person who's coming for genetic counseling or to see a physician, then what they want to know is that, hey, what is my risk of developing disease X by age 70? So they want to know what their risk of developing disease is. That's an absolute risk. Each person want to know. But if you just want to know, broadly speaking, yeah, compared to the people, you're twice as likely to develop a disease X as opposed to not developing disease X, because you have certain risk factors. So I think that's a really important distinction that one has to make. There are genes that show a drastic aging, and these are progeria conditions. So Werner syndrome is having one mutation in Werner's gene on chromosome 8 short arm can lead to a rapid aging, and there's a condition called Hutchinson whatever disease, so that you know somebody who's 5-year-old can look like can have a bold, but those people will have senility even before they reach age of 10, and then by 16 they usually die of heart disease. So that's an extreme case. So the other case is Down syndrome, where having 3 chromosome on 21, by the time, because they have 3 copies of gene called APP, by the time they reach age 40, they have all the neuropathological evidence of Alzheimer's disease, having plaques and tangles in their brain by their 40s. And by 60, they have more than 50% of them will have developed Alzheimer's disease. So it could be a single gene, or it could be multiple genes all working together. But the examples that I gave you are usually extreme cases, such that the likelihood of you observing them is very low. And the other study that I showed you in the earlier slide of healthy aging, where if you have a FOXO3A, you are more likely to survive to old age without major chronic diseases. So there are people who are studying these extremely old cohorts to see what makes them special. Is it biologically, or is it social network, or whatever, to see the protective factor that lead to healthy aging. So I believe that part of research is quite active. And then I have one study on healthy aging, and NIH is supporting a big consortium study to put together data of people who survived to 100 and older. So, to answer your question, we don't know, but it's definitely worth looking into. But you have to think about that. One of my earlier slides that says that if everybody lives to 100 years old, then as you've seen that in graph, that they're going to be experiencing all kinds of late onset chronic diseases, and those are going to create a large public health burden on the population. So there are a lot of strengths and weaknesses, but the bottom line is that we actually need to know what the nature is, and then how we can tackle those problems. So I think that's why I said research is needed, and it should be done in Guyana and for Guyana population.
Yes, thank you. Right, I think I will have to read my slides. Unfortunately, I don't know for what reason the system is not allowing me to share it. But I will read it. Now, so my talk is entitled, and thank you, Dr. Lee, and Dr. Previous presented before. This session deals with genetics. I will be talking specifically, I think the two former speakers speaks about the role of genes in diseases in general. Particularly, I think Dr. Lee focused on Alzheimer. I will focus primarily on suicide, and what are some of the genetic factors that can contribute to suicide in the context of our environment. And so my talk is premised on a lot of literature that is out there that seeks to make a case with respect to the genetics of suicide. We are very much concerned that suicide of itself constitutes a public health issue, and it involves numerous biological, psychological, cultural, social, and family determinants. These behaviors, suicidal behaviors, range from suicidal ideation to suicide attempts to suicide completion. And this represents a fatal dimension of mental ill health. Evidence of involvement of genetic risk factors in suicidal behavior is well documented. Even though in the literature, there is a lot of to and fro with respect, there is still need for more evidence to really nail home the point of the genetic factor with respect to suicide. However, there are a number of studies to which we can refer to, which somehow will help to support the notion that genes play a role in suicide. And so those studies that we mentioned are family studies, twin studies, adoption studies, and also looking at the pathway of tryptophan metabolism that involves in the synthesis of serotonin. And so I want to, the aim of, as I said, of this discussion is to look at the genetic association in suicidal behaviors through the use of family, twin, and adoption studies, and looking at the polymorphism of the serotonergic system. The serotonergic system is a system or the pathway that is involved in the synthesis of serotonin from tryptophan. And so with respect to family studies, many of there are reports in the literature that there's a high degree in first-degree relatives compared to control. And this was in adolescent population. So the study found that first-degree relatives have a greater risk of committing suicide. There's also higher rates of suicidal behavior in relatives of suicide victims. So you have a relative who would have committed suicide, it's more likely that you will attempt or you will commit suicide. And also from a family standpoint, the literature also reported high rates of suicide in offspring of suicidal parents compared to offspring of non-suicidal parents. I just wanted to also point out that there might be a number of mitigating factors that may contribute underlying apart from genetic factors or the mitigating factors. However, one such mitigating factor could be grief. Where persons, let's say, there are several conditions which can result in grief and which can cause persons to commit suicide. However, some studies have also shown that rates of individuals who lost their parents to suicide were higher compared to individuals who lost their parents to homicide or unintended injury, right? And so rates of suicide in individuals who lost their parents were higher compared to individuals who lost their parents from a homicide or from an unintended injury. Again, making the point, suggestion that the trend of familiarity of suicide completion may be at least partially independent of the familiarity of psychiatric diagnosis. Now, continue looking and to make a case with use of family studies to drive home the point of the genetic suicide, that we also saw an increased rate of suicide attempts or completed in families of suicide completers and proband. You know what is a proband? A proband is the first person in any generation, in any family, that presents with a disease. And so they actually saw a high risk of relatives of probands as compared to persons who had no, families that had no history of suicide. Suggestion the existence of common suicide phenotype that includes both attempt and completion. We know that our phenotype is determined by our genotype. And therefore, familial transmission may be independent of that of psychiatric disorders, despite the fact that most suicide attempters, completers, have underlying neuropsychiatric diagnosis. And so, again, a lot of meta-analysis is usually done to extrapolate and to eliminate all the other underlying psychiatric disorders that may also contribute to suicide in order to make a case for the genetic factors. With respect to twin studies, in the case of twin studies, comparisons were done between monozygotic twins and dizygotic twins. And the evidence, assuming that the environmental factors are similar between the monozygotic and the dizygotic twins. And several studies reported of higher rates in monozygotic twins in respect to dizygotic twins for suicide thought, suicide attempts, as well as completion, where there was a high rate in monozygotic twins, suggesting that suicide attempts and completion may share a common genetic component. In respect to adoption studies, several studies have shown that, again, there is an increased risk in persons who, because really and truly, when you're adopted, you change your environment, and so, but you maintain your genotype, and so studies have shown that persons, studies reported the risk of suicide in full siblings of adoptees who died by suicide before age 60 was significantly higher than in full siblings of adoptees who do not die by suicide. And so again, there is this strong genetic component as we look at family studies, twin studies, and adoption studies. Now, another factor that is considered in trying to understand, and this particular lecture, as I said, I'm doing it because even as we contemplate our trauma center, our research center, what are the kinds of research that we can do? As Dr. Lee indicated, here in Guyana, we need to do our own research. And so this whole concept is for us to contemplate and to look at the kinds of research that we can do so we can have our own understanding as to what are the mitigating factors, genetic factors, environmental factors, combination of the two that may be responsible for suicide in our environment. Now, another important aspect in trying to understand the genetics of suicide has to do with the serotonergic system, which is involved in the pathway in the development of tryptophan. It's unfortunate that you cannot see, but several studies have implicated altered serotonergic pathway in the pathophysiology of suicide behavior. And this has been a prime target for genetic studies of suicidal behavior. There has been evidence that low cerebrospinal fluid concentration of the serotonin metabolite 5-hydroxyacetyl acid has been found in numerous studies of suicidal patients. And so my slide here speaks about just a revision for those of the structure of the gene. You know, we have the promoter region, we have the coding regions, the non-coding regions, we have the flanking regions. And so just to, because in trying to understand the pathway, the serotonergic pathway with respect to how it can influence suicide, we just need to have a revision of what the gene structure looks like. The coding region, which is the exon, the non-coding region, which is the intron, and then we have the promoter region, which gives an indication as to where transcription begins. Now, the serotonergic system, again, as I go through this, it speaks about the role of tryptophan, the amino acid tryptophan, which is a precursor for the synthesis of melatonin, the synthesis of serotonin. Now, in the first step, tryptophan is hydroxylated using the enzyme tryptophan hydroxylase to L5-hydroxytryptophan, which is then decarboxylated using aromatical amino acid decarboxylase to produce serotonin. Serotonin, which is 5-hydroxytryptamine, right? Which is, and for this purpose of this presentation, we can refer to it as 5-HT. Now, the degradation of serotonin can be degraded by the monoamino oxidases. And you know, these are a group of enzymes that are really targeted by the antidepressant drugs to 5-hydroxyindole acetaldehyde. And so we have here tryptophan bring the precursor for the synthesis of serotonin, and the main enzyme, the rate-limiting enzyme, is tryptophan hydroxylase. And we also have the degradation of serotonin is by the enzyme, the monoamino oxidase. Now, several studies have done and have targeted polymorphism of that enzyme. That is the serogenic pathway. And so the enzyme, as we mentioned, which is tryptophan hydroxylase, there are several reports or several studies have done to show that there is polymorphism. Now, what is polymorphism? Polymorphism really means a different form of the gene. The gene can be represented in different forms. You have a variant allele apart from the normal allele. So within a population, you can have different genetic structure, different alleles within a population. And for a gene to be considered polymorphic, it must have a certain percentage in terms of representation. And so what we're really saying here is you have different types of the tryptophan hydroxylase among different individuals. So you have a genetic difference variation among the enzyme, the tryptophan hydroxylase, which is the main enzyme that is involved in the serotonergic pathway which converts tryptophan to serotonin, and serotonin, we know, which is responsible for mood, for a feeling of anxiety and everything else. And so we have variation or polymorphism of the tryptophan hydroxylase as one of the genetic factors which can contribute to different behavior. So as we said, that the two genes that code for the tryptophan hydroxylase are referred to as tryptophan hydroxylase 1 and tryptophan hydroxylase 2. Tryptophan hydroxylase 1 is expressed in the central nervous system and the periphery, whereas tryptophan hydroxylase 2 is expressed in the brain. By the way, tryptophan hydroxylase is located in chromosome 11, P5.3, P14. Now, there are several polymorphs or several types of variation or variants of the tryptophan hydroxylase 1 gene. One is referred to as the A218C. A218C, we have the A779C, and we have the A6526G. Those are the three variants. So you have a percentage of the population may have the gene A218C that codes for tryptophan hydroxylase. Another percent of the population may have A779C that codes for tryptophan hydroxylase. And we have another percent of the population may have A6526G. Now, what studies have shown that polymorphism in the A218C has been associated in attempters compared to non-attempters, right? So you have a higher level of suicide in this variant as compared to persons who did not commit suicide. And particularly, this was seen in age persons older than age 65. Now, the mutation there occurs at the level of the transcription factor. That's where we have the mutation or the variation. Now, with respect to A779C, they have seen which is located in the intron 7. That's where we have the variation at the level of intron 7. And so studies have shown that persons with this form of polymorphism are associated with attempt suicides or those who have committed suicide or attempted previously. Either you have a past experience of attempting suicide or currently trying to commit suicide. And also to increase susceptibility to psychiatric disorders in general, which in turn are characterized by increased incidence of suicide. So again, you have some other psychopathology that can be underlying factors that may also contribute. And that's why there's so much of confusion in the literature, with respect to isolating the genetic factors, because sometimes there seems to be other psychopathologies which can also contribute to suicide. Now, with respect to the other polymorphic form of the tryptophan 1 gene, which is A6526G, and this has been located in the promoter region, and this has been associated with suicide attempts. And this variant, according to the literature, seems to be independent of gender and other psychopathology. So, in other words, what they're saying is that persons who manifest this gene, this polymorph, with respect to the tryptophan hydroxylase, have a greater risk, or the genetic factor can be considered independent as compared to the other forms which may have other mitigating factors. That's the A218C as well as the A779C.
Excuse
No.
me, sorry, Dr. Cummings.
Yeah.
I'm sorry to interrupt, but if you can maybe wrap up a little bit. We have ran over time, and we have a next talk soon. Thank you.
Well, I'm just, there are other factors which I wanted to discuss. Okay, well, I would just say clearly that the tryptophan 2 gene has also been associated with suicide completion. Studies have shown that there is an overexpression of this form of the tryptophan H2 gene, the variant, in persons with depression and suicide. Another factor is the serotonin transporter gene variation. In this one, there are 2 types of polymorphism that have been, or 2 forms that have been isolated with respect to serotonin transporter, which again associated with some form of suicide. And finally, we have the monoamino oxidase, and that's the enzyme that's responsible for degrading serotonin into the acetaldehyde. And there seems to be some indication that this enzyme, which we know that is targeted, as I mentioned earlier, by the antidepressant drug, there seems to be some elevated activity of the monoamino oxidase in the hypothalamic region of suicide victims. So I will have to skip a few slides and conclude that genes really work alone, that the mode of inheritance which is from a genetic standpoint, that may contribute to suicide could be polygenic, could have several genes. As other speakers mentioned, that suicidal behavior is very complex and may involve environmental factors. Other mitigating factors can be childhood trauma, epigenetic factors. Our first speaker speak about we'll talk within epigenetics. Epigenetic factors may also play a role where there is no change in the genome of itself, but the environmental factors can change in the expression of the gene. And to conclude, finally, that genetic factors do influence the risk of suicide, probably independently of other psychiatric disorders. And so the literature out there is still very young. The literature, but there is some attempt to link genetic factors to suicide, and a lot of it has to do with trying to understand the metabolism, the synthesis of serotonin, which is important in our behavior, and to see how variation with respect to the genes that are responsible for the enzymes that are involved in that pathway, where variation in the genetic makeup of those enzymes, how they can contribute to suicidal behavior. Thank you very much.